Hi, my name is Scott and welcome. This presentation is on metabolism, and more specifically, it's on the metabolism of nutrients to energy. Basically, what we're going to look at here is how we convert the foods that we consume into energy and how we harness that energy so we can spend it when we need it in order to maintain our life. Now, this is the metabolism lecture for HUN 1201, the science of nutrition, or some colleges and universities may call it principles of nutrition or essentials of nutrition. This will meet those needs for that class as far as the metabolism lecture. This is one of my lectures for the lecture series, HUN 1201, that you can see at my website at mydietandnutrition.com. So let's get started. So the way we receive our nutrition, human beings, we eat either plant or animal, and some of us eat animal foods. And somewhere down the line, those animals have eaten plant sources also. So we receive our energy from these two food sources. And the way plants will receive energy is they take in sunlight as a form of energy. And they will take in carbon dioxide and water. And they will give us carbohydrate in the form of glucose and release oxygen. So for every 6 molecules of carbon dioxide plus 6 molecules of water, we will get 1 molecule of glucose and 6 molecules of oxygen. And that's where we get our energy from plant sources and from animals that eat plant foods also. A brief term is metabolism, the combined sum of all the chemical reactions in living organisms that occur at the cellular level to maintain life. There are 2 phases of these chemical reactions, that is anabolism and catabolism. Anabolism is the process in which energy is consumed to build bonds of smaller molecules such as amino acids to form larger molecules such as proteins. Also monosaccharides to form polysaccharides and amino acids to form polypeptides, proteins. Catabolism is the process in which energy is liberated from the bonds as larger molecules such as glycogen and polypeptides and triglycerides are broken down into smaller molecules such as glucose, fatty acids, glycerol, and amino acids. Okay, nutrients to energy. Basically, all the foods that we consume will be broken down into these 3 nutrients, and that is carbohydrates, proteins, and fats. Carbohydrates will yield glucose. Polysaccharides will be broken down into yield, and also glycogen will be broken down to yield glucose. And glucose is a 6-carbon molecule. The proteins yield amino acids, which contain 2 or more carbons. And this is a representation of arginine. It has 6 carbons. And fats will yield, the triglycerides will be broken down into a glycerol backbone and a fatty acid. And here I have a picture of palmitic acid or palmitate. And this is 16 carbons long. Now, the way we get our energy from these nutrients is there is energy stored in the bonds. This carbon to hydrogen bond, carbon to oxygen, and carbon to carbon bond. The carbon to nitrogen bonds, and the carbons to oxygen. All these bonds contain potential energy. So when we break these bonds, that liberates energy. And then we're going to take that energy and harness it in the form of ATP. And this is a molecule of ATP. This is adenosine triphosphate. And this is what we use as our form of currency. ATP is considered the body's form of energy currency. So ATP contains high energy bonds. These are 3 phosphate bonds. There's 2 bonds here, and these in between these phosphates are high energy bonds. So the breakdown of adenosine triphosphate, basically it is adenine, which is a nucleobase, such as in DNA, and there is a ribose. It's a 5-carbon sugar, and it contains 3 phosphates. So adenine and ribose together is called adenosine, and this is adenosine triphosphate. Now the way we hydrolyze this is we add water, and then we'll break the bond of the phosphate, and that will yield energy. It will liberate some free energy. So ATP, it contains these bonds that are cleaved off by enzymes ATPase. And the phosphates are bonded together by the enzyme ATP synthase. So oxidation reduction. Now this is a concept that I really need you to get because we're going to carry it through this whole presentation. Some people have a little challenges get it, but what I've done is I'm going to break it down into 4 segments. But before we get to that, I want to explain electronegativity. And this is a concept I need you to understand. Basically, this is the periodic table of elements, and all these elements are in their pure form. These pure atoms have a 0 charge because the number of protons are positive, and the electrons, which are negative, they're equal. So like I said in previous lectures, the nucleus of an atom has protons and neutrons. The protons are positive, the neutrons are neutral, and the electrons that rotate around the nucleus are negative. So all these atoms have an equal number of protons and electrons, so they have a 0 charge. Now if we look at this line that I have here, this is the increasing of electronegativity. And electronegativity is just the ability of an atom or molecule to attract electrons. Now, for example, this is francium. This was found in France, so they called it francium. This is the least electronegative on this periodic table. And fluorine is the most electronegative. So fluorine will draw the electrons from francium to fluorine. Actually, francium is a radioactive isotope, so that's probably a bad example. So let's use oxygen and nitrogen. Oxygen is more electronegative than nitrogen, and oxygen will draw nitrogen's electrons to it. So let me give you an example of that. Say, for example, this is a water molecule, and oxygen, if we look at oxygen up here, oxygen has a total of 8 electrons, but it has 6 electrons in its outer orbital, the outer balanced shell. So here are the 6 electrons, and normally you'll see oxygen with 2 lone pairs. This is a lone pair, and this is a lone pair, which kind of push these unpaired electrons down to an angled or bent shape. And then what they will do, these unpaired electrons will bond with other atoms. And in this case, water is going to bond with hydrogen. Hydrogen, if we look at hydrogen, it has 1 electron, and it has actually 1 electron totally in its valence shell, so it has 1 electron. So oxygen's unpaired electron will bond with hydrogen. Now, since oxygen is more electronegative than hydrogen, it will dominate hydrogen's electron. So hydrogen's electron will spend more time orbiting oxygen than it will hydrogen. And it will dominate that electron. And by dominating that, it will add 1 electron to oxygen. So now the charge on oxygen is not equal anymore. It has added an electron, which is a negative charge. So oxygen with 1 hydrogen will become a -1. And this is the hydrogen, and this is the other hydrogen. So oxygen will become -1 charge when it dominates this 1 electron because of its electronegativity. And when it dominates this other hydrogen over here, it will have 2 electrons that have negative charges. Now, oxygen originally had a 0 charge, but with 1 electron and another electron, each are being negative, that will have a -2 or a 2 minus a charge. Now, the hydrogens, since they are losing that electron, and now since they're losing 1 negative electron, they will have 1 more proton in that atom, so that will be a +1. So it has 1 more positive charge. And likewise for the other hydrogen. So we're going to come back to this, and this is atomic numbers, so if you will. And so leaving that, let's go to the oxidation reduction. This is a concept that I need you to get. 1 second. Now, in the past, before we recognized the electrons, that was before Sir Thomas. It was a British physicist who identified electrons somewhere around the 1890s. Before then, we used to refer to oxidation reduction. We used to refer to oxidation is the gain of oxygen, and reduction is the loss of oxygen. Now, that was somewhat of an archaic term, and then when Sir Thomas recognized the electrons, then we became into a greater understanding to where we recognized that oxidation, now we're going to use all 3 of these throughout this presentation, but then when we recognize the electrons, we realize that oxidation is the loss of electrons and reduction is the gain of electrons. Let me give you an example. Sorry, I forgot about this. Let me give you an example of oxidation is the gain of oxygen. Here we have carbon and 2 mercury oxide molecules. Now, if oxidation is gain of oxygen, carbon will actually gain that oxygen. So carbon is oxidized. And the mercury oxide, it will lose its oxygen, so the mercury has been reduced. So that's the old term. And now with oxidation is the loss of electrons and reduction is the gain of electrons. Let's do an example of that. This is 2 molecules of magnesium plus a dioxide. Now, let's go back to the table of elements and let's look. Magnesium is less electronegative than oxygen. So magnesium, since it's less electronegative, oxygen will pull those 2 electrons from magnesium because it has 2 electrons in its valence shell. So it will draw those 2 electrons to oxygen. Now oxygen will have a 2- charge, and since magnesium has lost the electrons, magnesium has become oxidized and oxygen has become reduced. Now, to further this, in biochemistry and nutrition biochemistry, we tend to look at hydrogens a little more. It makes it a little easier. So oxidation is the loss of hydrogen, and reduction is the gain of hydrogens. So let me do an example of that. This is methane and 2 molecules of oxygen. Dioxide is a diatomic molecule of oxygen. Now the carbon will lose all of its hydrogens and it will gain an oxygen. Carbon will lose all of its hydrogens. So since carbon lost its hydrogen, the carbon has been oxidized. And the oxygen will gain all the hydrogens. So the oxygen has been reduced. And it goes with the electronegativity. Oxygen has pulled the hydrogens from the carbon because it is more electronegative than the carbon. Now, with the oxidation number, we did that with the molecule of water with oxygen having a 2- charge as it takes the 2 electrons from hydrogen. So let me give you an example here that I'm going to use later in the electron transport chain. Now this is iron at its 3+ state. That is ferric iron, and iron at its ferrous state. Now the oxidation number, if we look at the more positive number, then that is the one that has been oxidized greater than this, because this ferrous state has 1 more electron than this ferric oxygen. So we can look at the oxidation number to determine also which one has been oxidized. Now when we get to the electron transport chain, I'm going to expand on this concept of oxidation reduction with another term. So, and it's something that always plagued me all the way through undergraduate school, is whenever you gain electrons, why is it said to be reduced? When you gain hydrogen, when you gain something, why is it reduced? That kind of plagued me all the way up to my graduate year, and then finally it was answered. So I'm going to share that with you also. Okay, a couple definitions: coenzymes and cofactors. Coenzymes are organic molecules, normally proteins, that contain a vitamin or a mineral that are required by certain enzymes to act as a catalyst. And catalysts help facilitate a substrate to a product. They bind to active sites of the enzyme and help facilitate a reaction. Coenzymes often function as intermediate carriers of electrons, and we'll see how this works in an electron transport chain. Some examples include FAD, which is flavin adenine dinucleotide, and NAD, which is nicotinamide adenine dinucleotide, and coenzyme A and coenzyme Q. This is the oxidized form of flavin adenine dinucleotide, and this is its reduced form. This is the oxidized form of nicotinamide adenine dinucleotide, and its reduced form. Now, flavin adenine dinucleotide has a part of its structure is riboflavin, and that's vitamin B2. And part of nicotinamide adenine dinucleotide structure is niacin, and that's vitamin B3. And coenzyme A, part of its structure is pantothenic acid, or pantothenate, and that's vitamin B5. And we're going to go over each one of these structures coming up. And coenzyme Q. So cofactors are normally considered inorganic substances that are required to help facilitate a reaction also. And some of those examples include zinc, iron, copper, potassium, and magnesium. We're not going to go in depth with this two cofactors. That's at a higher level than we're going to go right now. So let's cover first, as you just heard, this is a dinucleotide. So let's define what a nucleotide is first before we go any further. Now this is a nucleotide, and a nucleotide is composed of three items. It's a nucleobase. This is a nucleobase. And these are nucleobases that constitute our DNA or RNA. And it can be either one of these. This here is adenine. And it also is composed of a 5-carbon sugar. This is a 5-carbon sugar, and this is ribose. And it also contains one or more phosphate groups. So this is a phosphate. So it contains a nucleobase. Also a nucleobase is a 1 or 2 ring structure, a nitrogenous structure, which has a nitrogen in it. And in this case, again, it's adenine. And just a little bit further, DNA, guanine will pair with cytosine, and adenine will pair with thymine. And in RNA, guanine will pair with cytosine, and adenine will pair with uracil. So that's a nucleotide. And again, like I was saying, these have two nucleotide bases, two nucleotides. This is the coenzyme flavin adenine dinucleotide. It has two nucleotides. And let's break this structure down. This is one nucleotide. This is adenine. It has its nitrogenous base. This is the ribose sugar, and it has a phosphate. And the other nucleotide is right here. This is an isoaloxazine ring, but this structure right here is actually uracil. So this is the nitrogenous base. It has a 5-carbon sugar and a phosphate. So it has two nucleotides in it. Now, in addition, this is where the flavin adenine dinucleotide will be reduced. So this will oxidize a product where flavin adenine dinucleotide will reduce itself. And this is one of the points where it will be reduced, and this is the other point where this will reduce. Now, like I was saying, part of this structure is vitamin B2. It is riboflavin, and here is the structure of riboflavin. This is riboflavin here. It has this isoaloxazine ring, and this ring is what gives riboflavin its yellowish color. Now, it has this ring, and it also has this 5-carbon sugar. And what we've done here is we just simply reduced this compound where it will break this oxygen bond and add hydrogen to it. And there we have ribitol. And ribose will go to ribitol simply by reducing this 5-carbon sugar. And there's the ribitol. Okay, so like I was saying, this will be reduced here, and that will be one hydrogen. Let me go back here. This will be one hydrogen whenever it will turn into the reduced FADH, and then with the other hydrogen, it will be FADH2. Okay, so nicotinamide adenine dinucleotide. This is its oxidized form, and this is its reduced form. So this is the structure here. This is its oxidized form here, and this is the reduced form here. As you can see, the hydrogen here has been put on at this point here to where it is reduced. So let's break this structure down again. This is the nitrogenous base, and this is a pyridine. A pyridine is a derivative of a pyrimidine. And if we go back one second here, these are the nucleotide bases, the nucleobases. And these are purines with a 1-ring structure, and these are pyrimidines with 2 rings. And a derivative also of this pyrimidine is a pyridine, and that's where nicotinamide comes in. So let's go back to that. And this is the nucleobase here, and this is very, very similar to niacin. This is the structure that will be, that I was saying that is considered to be niacin. Now the difference here is nicotinic acid has a hydroxyl here instead of having the amine group put on at this point here. So this is where we see niacin in nicotinamide adenine dinucleotide. And here is the 5-carbon sugar base and its phosphate. Now this is the other nucleotide, and this is adenine, this is the ribose sugar, and it's phosphate. And again, this is where it is reduced right there. Okay, cellular respiration. As we saw in photosynthesis, the plants will yield a glucose molecule and 6 molecules of oxygen. And through our process of metabolism, we will yield 6 waters, 6 carbon dioxides, and in between 36 to 38 ATPs, depending upon what tissues are respirating this. And then we'll also be giving off heat in the cellular respiration. So, excuse me. So cellular respiration is a set of metabolic reactions that take place in cells, which constitutes 3 biochemical pathways. That's glycolysis, the tricarboxylic acid cycle, and the electron transport chain. These are processes by which cells convert energy from chemical bonds of nutrients into adenosine triphosphate and release waste products. So these processes are carried out the glycolysis is carried out in the cytosol of the cell. Now this is the mitochondria, and around this is the cytosol. That's the aqueous environment of the cell. And glycolysis is carried out in that aqueous environment. The tricarboxylic acid cycle is carried out in the matrix. This big open space in the center of the mitochondria is the matrix, and that's where the Krebs cycle, citric acid cycle, or the tricarboxylic acid cycle is carried out. It has a few different names. And the electron transport chain is carried out in the inner membrane, and this is the inner membrane. It has an outer membrane, and it has an inner membrane, and the electron transport chain is carried out inside that. Okay, a general overview. Say if we eat a slice of pizza, it will be broken down into proteins, carbohydrates, and fats. Now, the carbohydrates will be broken down to a glucose molecule. And then glycolysis is the process from converting glucose to pyruvate, a 6-carbon molecule, into 2 3-carbon molecules of pyruvate. And fats will be broken down into the glycerol backbone and fatty acids. And protein will be broken down to their amino acids. Now, what I'm going to point out here is the junction between pyruvate and acetyl-CoA. This is like an interchange on a highway. We have many different things coming in and out, and I want to point out what can go where. So pyruvate can go to acetyl-CoA and go into the citric acid cycle, creating hydrogens and electrons to be shuttled to the electron transport system. And pyruvate can also go back to glucose, but it is not a reversal of glycolysis. It is actually gluconeogenesis. Gluco meaning glucose, neo meaning new, and genesis is the creation of new glucose. And that process takes several different substrates and enzymes, so it's not a reversal of glycolysis. So, but acetyl-CoA, once pyruvate goes to acetyl-CoA, it is committed and it can't go back to pyruvate. Now fats, the glycerol can go into pyruvate, and it also can be converted back to glucose. Fatty acids can go to acetyl-CoA, and acetyl-CoA can be converted into fatty acids. Pyruvate can also be made into amino acids, and amino acids can be made into pyruvate. And amino acids can go into acetyl-CoA. There are some amino acids that can go into pyruvate, and some that can go into acetyl-CoA. And there are amino acids that can go straight into the tricarboxylic acid cycle and yield products to go into the electron transport chain. Now, also pyruvate, through a process of fermentation, can go into lactate. Say if we're exercising and we're sprinting or something and our cells run out of oxygen, that will cause pyruvate to be fermented. And normally when you ferment a sugar, you will yield alcohol, an acid, or some type of gas. But here, pyruvate will yield lactic acid. And this will happen only a few minutes, and then we'll start to run out of energy there. So again, the tricarboxylic acid cycle and the electron transport chain is in the mitochondria, and that's aerobic, that is with oxygen. And the glycolysis is in the cytosol, and that is without oxygen. Okay, glycolysis. Glycolysis comes from the Greek stem word, the stem glyk, meaning sweet. And lysis means dissolution. So glycolysis is sweet dissolution. It's the dissolving of a sugar. Okay, glycolysis is glucose to pyruvate. It is the sequence of reactions that metabolizes one 6-carbon molecule of glucose to two 3-carbon molecules of pyruvate. 6 carbon molecules of glucose to 3-carbon molecules of pyruvate. It is an anaerobic process without oxygen that occurs in the cytosol. Now this is the whole overview of glycolysis. It looks a little bit intimidating right now, but I'm going to break this down in the next 2 slides into phase 1. This is phase 1 here. This is the investment phase. This is where it costs you 2 ATPs. And then on the second slide, I'm going to break this down into the payback phase, is where you'll gain 4 ATPs. So you will lose 2 and gain 4, so you'll have a net gain in glycolysis of 2 ATPs. And then you'll also get some nicotinamide adenine dinucleotide here be reduced. So let me, like I said, I'm going to break this down from here. Glucose will go into this fructose 1,6-bisphosphate, a 6-carbon molecule. And then from that, it will be split into 2. So that will be the second slide. So let's start with the glucose. And glucose is going to be phosphorylated. This enzyme, hexokinase. Now, kinases participate in phosphorylation. So hexokinase is going to phosphorylate. It's going to take one of ATP's phosphates and put it on this glucose molecule. And it'll put it on the 6 carbon. 1, 2, 3, 4, 5, 6 carbons. So here you have, now you have glucose 6-phosphate. This has been energized. Now we have, in the second step, and there's 10 steps in glycolysis. The second, and we look at these steps at the enzymes that help facilitate that reaction. So phosphoglucose isomerase, what this is going to do is just going to rearrange this 6-carbon cyclic hexose sugar into a pentose sugar, a 5-carbon cyclic pentose sugar. So basically what's going to happen is this first and second carbon are going to do a little rearrangement. So you'll have a 5-carbon cyclic structure, and then you'll have the 6th carbon coming off as a substituent. Then the third step is phosphofructokinase, another kinase, and it's going to cost an ATP here. Well, that will put on another phosphate, and that will become fructose 1,6-bisphosphate. So now you have a phosphate on number 1 carbon and on number 6 carbon. Now this is a very unstable molecule, and what's going to happen is we're going to have to split this in half. So aldolase will split this in half, and then you'll get 2 3-carbon molecules. And one of them is dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate can go to pyruvate, but this dihydroxyacetone is unable to go to pyruvate. So we're going to have to change this around a little bit and make it another glyceraldehyde 3-phosphate. So everything at this point now, we'll use triphosphate isomerase to change this molecule around to get the second glyceraldehyde 3-phosphate. So everything now from this point is going to be doubled. We have 2 glyceraldehyde 3-phosphates coming in right here. So now we have 2 glyceraldehyde 3-phosphates, and everything will be doubled. So once we have an NAD come in, it's going to be doubled. Everything will be doubled from this point because we have 2 molecules we're working with, and the end result is going to be 2 molecules of pyruvate, a 3-carbon molecule. So here we have glyceraldehyde 3-phosphate. And the 6th step, this enzyme, glyceraldehyde 3-phosphate dehydrogenase, is going to take off some hydrogens. And it will take off those hydrogens. It will be this G3P, they call it short, glyceraldehyde 3-phosphate, it will be oxidized. And NAD, its oxidized form, will be reduced. And we'll also add a couple phosphates in there. And for each one of these, for each one of those, we'll add a phosphate to each one of these. And we'll end up with 1,3-bis phosphoglycerate. And the bis means 2 phosphates. So now we have 2 phosphates at 1 and the 3rd carbon. Now we're going to do a phosphoryl transfer with a phosphoglycerate kinase at step 7. We're going to take off one of these phosphates, and Now this is where we start to get back some energy. So ADP will come in here and it will come out as ATP. We're taking off one of the phosphates here and gaining an ATP. Now step 8, phosphoglyceromutase, we're going to just rearrange this atom here. 2 of these, we're going to change the phosphate over to the second carbon. So it's just a mutation of this, and we'll move that carbon over. Now we have 2-phosphoglycerate, and then the 9th step, enolase, we're going to take out a molecule of water for each one, so that will be 2 molecules of water. And that way we are taking out a couple hydrogens and an oxygen, and that way we're ending up with a double bond. So now in step 10, that will leave us with phosphenylpyruvate. They shorten this for PEP, phosphenylpyruvate. Now for step 10, pyruvate kinase, another kinase, is going to do a phosphoryl transfer. And we're going to take this phosphate off of PEP, and now we will have pyruvate. So we gain another ATP at this point. So here we're gaining 2 ATPs for each molecule. So we have a total of 4 ATPs in this payback phase. And here, right after glyceraldehyde 3-phosphate, we're reducing NAD plus to NADH. So we'll have 2 of those. Now for every NADH, the electron transport chain, can make 3 ATPs. For every 1 NADH, we make 3 ATPs. And for every 1 FADH, we only make 2 ATPs. So with that being said, we have 2 molecules. The end result, we take a 6-carbon molecule of glucose, and our end result in glycolysis is 2 3-carbon molecules of pyruvate. And the net gain in glycolysis is 2 NADHs, 2 ATPs, 2 molecules of water, and 2 pyruvate molecules. Now, pyruvate to acetyl-CoA. This is the intermediate point in between glycolysis going into the citric acid cycle. And this is one of the junctions where we have nutrients coming in or going out that can be converted or not. So this is the pyruvate molecule, and it has to go into the mitochondria. Once it's in the mitochondria, then it will be decarboxylated. We'll take off this carbon and the 2 oxygens, and it will come out as carbon dioxide. It will also be reduced. Pyruvate will be oxidized, and NAD plus will be reduced. So nicotinamide adenine dinucleotide will oxidize pyruvate, reduce itself, and then we'll have a coenzyme A be attached right here. As this leaving group comes off, coenzyme A will be attached, and now we have acetyl-CoA. So the 3 steps here, this is done with the enzyme pyruvate dehydrogenase complex. It's a 3-step process, and it's a very big complex. And we're not going to go over that at this point, but again, the 3-carbon pyruvate is decarboxylated, losing the carbon dioxide. The 2-carbon anion here will then lose 2 electrons, and that will reduce NAD. And then the coenzyme A is transferred to this 2-carbon cation, forming the acetyl-CoA. And the oxidation form of NAD plus is reduced by pyruvate dehydrogenase complex, forming the NADH. Okay, so this is what a coenzyme A looks like. This is the coenzyme A. And here we have a nucleotide. This is adenine again, and this is the ribose sugar and the phosphates. Now, like I said, coenzyme A contains the vitamin B5, pantothenic acid, or pantothenate. And this is pantothenate right here. And it also contains beta-mercaptoethylamine. Now, this is the amine, and this is the ethyl, 2 carbons. And mercapto basically is a sulfur with the hydrogen. It's a sulfur atom. So that's mercaptoethylamine. Now, this is the coenzyme A. And what we're going to do, now that we have the acetyl built in the last step, this acetyl will bind to this sulfur. And oftentimes you'll see this as simply the acetyl-CoA. You may see the SH on there, or you may just see the S, but this is what you'll actually see. It will bind right here as this needs to bind with the electron here. So acetyl-CoA, again, is the link between glycolysis and the citric acid cycle. So another way we can make energy, again, pyruvate can go to lactate, and this is called the Cori cycle. Glucose in the muscle will go around lactates to glucose, and this is the Cori cycle. Pyruvate accepts hydrogens from glucose under anaerobic conditions when you don't have oxygen. When you're sprinting for a few minutes and your cells run out of oxygen, then that lactate will be fermented to be lactic acid. So under anaerobic conditions, or when there's insufficient mitochondria, and that will become lactate. So the glucose will go through the glycolysis to pyruvase, but you won't have the oxygen. So it will go through the lactate dehydrogenase complex, and then NADH will be oxidized, and the pyruvate will be reduced, becoming to lactate. And then that's in the muscle. And then this will go to the liver, and those lactates, again, will be converted from lactate dehydrogenase complex. The NAD, the oxidized form of NAD will be reduced, and then the lactate will be oxidized, becoming pyruvate again, and then the pyruvate through gluconeogenesis will become glucose, and then that can go back into the muscles and the tissues. So this is the Cori cycle, and here is where the nicotinamide adenine dinucleotide is oxidized and pyruvate is reduced. So here the hydrogens are coming onto pyruvate, and here is lactate, which is the reduced form of pyruvate. Okay, the junctions of pyruvate and acetyl-CoA. Here again, the junctions of pyruvate and acetyl-CoA, these are very interesting. These are like, again, the interchanges on a highway. So glucose can go to pyruvate, like I said, and through gluconeogenesis, not a reversal of glycolysis, can go back to glucose. Now, when fatty acids are broken down and you yield the glycerol backbone in the fatty acids, the glycerol can come in to make glucose, or it can go into pyruvate. And then pyruvate can go down to acetyl-CoA, into the citric acid cycle, and to the electron transport chain. The fatty acids, they go into acetyl-CoA. When fatty acids are broken down, they're broken down at 2 carbons at a time, and we'll go through that coming up. And acetyl-CoA, remember, is the 2 carbons. So acetyl-CoA, since it's 2 carbons, you can keep building the 2 carbon units and make fatty acids. And there are amino acids that are ketogenic, meaning they will come in at acetyl-CoA, and there are amino acids that can go into pyruvate. And these are glucogenic, meaning they can make glucose, and these amino acids can actually make fatty acids. And then the lactate, again, like I said, pyruvate can go to lactate, and lactate can go back into pyruvate. So that's the overall look at the junctions of pyruvate and acetyl-CoA. But acetyl-CoA cannot go back to pyruvate. It's the committed step to go either into the citric acid cycle or possibly make fatty acid. The citric acid cycle. This is a graphical representation of the citric acid cycle. It is cyclic in that its 4-carbon molecule, oxaloacetate, will go through this cyclic molecular process and end up back to oxaloacetate. It's a 10-step process, and we identify those steps through the enzymes that helps facilitate that reaction. I've eliminated 2 of the steps, so I'm going to go through 8 steps, and I'll point out the 2 steps that I've simply eliminated. You'll often see it like 8 steps or sometimes 10 steps. So if you remember, pyruvate is the ending product of glycolysis. Pyruvate will enter the mitochondria and be converted to acetyl-CoA. So the first step, citrate synthase, will help transfer 2 of the carbons from acetyl-CoA onto the 4-carbon molecule oxaloacetate. By losing a coenzyme A, it is converted to citrate. And citrate is acidic because it has 3 carboxyl groups on it, which is where we get 2 of its names, the citric acid cycle or the tricarboxylic acid cycle. There's another name, the Krebs cycle, named after Hans Krebs. He was a Jewish German citizen who elucidated this mechanism, and he ended up fleeing Germany prior to World War II. So after citrate, we're going to have a dehydration. A water molecule is going to come out, yielding cis-aconitate, and then the water molecule will come back in to yield isocitrate. So basically, I've eliminated the dehydration and the hydration. The purpose is to get citrate into its isomer, isocitrate, which is also a 6-carbon molecule. And here we have the rearrangement of this citrate to isocitrate helps prepare this molecule for step 3, so we can dehydrogenate it and decarboxylate it. So here, the enzyme in the third step, isocitrate dehydrogenase, will help oxidize isocitrate into alpha-ketoglutarate. The oxidized form of nicotinamide adenine dinucleotide will be reduced as it takes off the hydrogens, converting the NAD to NADH. And we'll lose a carboxyl group off of isocitrate, and that will come out as carbon dioxide. Now we have the 5-carbon molecule alpha-ketoglutarate. Now in the fourth step, it is another dehydrogenation and another decarboxylation, where alpha-ketoglutarate dehydrogenase complex will decarboxylate. It will take off another carboxyl unit coming out as carbon dioxide, and it will take off a couple hydrogens through the process of the oxidized form of NAD, converting it to a NADH. Then that will yield succinyl-CoA. This is a 4-carbon molecule, succinyl-CoA. Now the fifth step, succinyl-CoA synthetase, is going to remove the coenzyme A, and that will liberate some free energy, where guanosine diphosphate will pick up the phosphate and add guanosine triphosphate through that energy. Now the sixth step, succinate dehydrogenase, is another oxidation process where we will oxidize succinate to fumarate, and flavin adenine dinucleotide will do the oxidizing of that as it is reduced to FADH2. And there we still have a 4-carbon molecule coming into the 4-carbon molecule fumarate. Now the seventh step, fumarase, will add in a water molecule onto the second and third carbon of fumarate, yielding malate. Now the eighth step, malate dehydrogenase, it will dehydrogenase malate by taking off a couple hydrogens and thereby creating another reduced form of nicotinamide adenine dinucleotide, yielding the oxaloacetate. Now the whole purpose of this citric acid cycle is to yield these coenzymes, or these intermediate electron carriers, that we can bring to the electron transport chain in the inner mitochondrial membrane. And the citric acid cycle will basically yield 30 ATPs. The first step in glucose, it cost us 2 ATPs. And whenever we converted glyceraldehyde 3-phosphate, we oxidized that and we reduced 1 NADH. So now we have, since it's 2 molecules of pyruvate, we have 2 NADHs. So we have a negative 2 ATPs, and we've gained 2 NADHs. Now, like I said, for every NADH, that's equivalent to 3 ATPs once it goes through the electron transport chain. So let's count some beans. 2 times 3 is 6. So that will give us 6 ATPs once we get through the electron transport chain. And that's coming up, but I just want to maintain the count of the ATPs while it's kind of still fresh. And the good thing about videos, you can go back and look at what I was going over. So in the payback phase of glycolysis, we gained 2 ATPs and then another 2 ATPs, and that was a total of 4 ATPs. But 4 minus 2 will give us a net gain of 2 ATPs in glycolysis. And the 2 plus the 6 is 8. Now, in some tissues, this nicotinamide adenine dinucleotide, it is going to cost us 1 ATP to get across the mitochondrial membrane. That's in some tissues, such as the brain and skeletal muscle. So that will cost 1 ATP for each NADH. So if you're having a total of 6, then you can reduce that by 2, and then it's 4, and that 4 plus 2 ATPs is 6. So that's where we come up with the difference between 6 to 8 ATPs in glycolysis, and it depends on the tissue. Now, once we got pyruvate into the mitochondria, we converted that to acetyl-CoA, and we were able to reduce the oxidized form of NAD and get 2 NADHs. So here we have 2, and again, one of these will equal 3 ATPs, so 2 times 3 is 6 ATPs. And everything, again, like I said, is doubled. So we had an NADH here, and you can go back and check this out. There's an NADH at each one of these points. So that's 2, 4, 6. 6 NADHs in total for each for 2 revolutions of this cycle, because there's 2 molecules of pyruvate. So that is 6, and again, each one yields 3 ATPs. So 3 times 6 is 18 ATPs. And this guanosine triphosphate is equal to an ATP. So there you'll get 2 ATPs there. And this FADH is equal to 2 ATPs in the electron transport chain. So here we have 2 of them. So 2 times 2 is 4. That's 4 ATPs. So the net gain in the citric acid cycle is 30 ATPs. And the total ATPs for these glycolysis and the citric acid cycle is 36 to 38. And the discrepancy here is because of these 2 that will cost us to get NADH across the mitochondria in some of the tissues. So that's counting the ATP. The electron transport chain, the chemiosmotic theory of oxidative phosphorylation. As I go through this process to yield adenosine triphosphates, I'll identify each one of these terms and what that really means. But first, the premise of the electron transport chain is the electron transport chain functions from a low reduction potential to a high reduction potential. The acceptor is the oxidant. NAD positive, the oxidized form of nicotinamide adenine dinucleotide, is an oxidant, and it has reduction or reducing, they both mean the same thing, potential. It has reduction potential or reducing potential. The more positive, the more the reducing potential the oxidant has to reduce itself. The donor is the reductant. NADH is a donor, and it has oxidant potential. The more negative, the more oxidant potential the reductant has to oxidize itself. Therefore, a reductant, the more negative oxidant potential becomes less negative. Oxidant, the more positive reducing potential becomes less positive. Therefore, the oxidized form of nicotinamide adenine dinucleotide to its reduced form is said to be reduced because, and this is what I was talking about earlier, is why is it when you gain electrons or gain hydrogens, is it said to be reduced? Well, the reduction potential of the free energy has been reduced. Okay, so this is a graphical representation of the electron transport chain. The electron transport chain is just a chain of protein complexes embedded in the inner mitochondrial membrane. And as we take these electrons from the reduced form of nicotinamide adenine dinucleotide, or the electrons from succinate, as it passes through these protein complexes, it will yield a molecule of water as these electrons are transferred. So that's why it's called the electron transport chain. Now, let me identify this structure. This is the intermembrane space, the inner mitochondrial membrane, where these protein complexes are embedded, and this is the matrix of the mitochondria. So this is complex 1. It's called NADHQ oxidoreductase. It's also referred to as NADH dehydrogenase. Complex 2 is succinate Q reductase, and it's also referred to as succinate dehydrogenase. Complex 3 is Q cytochrome C oxidoreductase, and sometimes you'll hear that referred to as cytochrome B C1 complex. Now, complex 4 is cytochrome C oxidase, and complex 5 is ATP synthase. Now, what we're going to do is basically we're going to follow 2 electrons from the reduced form NADH plus H through complex 1 to ubiquinone, complex 3, cytochrome C, complex 4, and then to build a molecule of water. So let's start with that. And the electrons pass from the reduced form will become of NADH plus H. This will become oxidized. And then the oxidized form of flavin mononucleotide, which is the isoalloxazine ring to be a nucleotide, is mononucleotide. The isoalloxazine ring, the 5-carbon sugar and the phosphate, that is a mononucleotide. This is its oxidized form. So when it takes the 2 electrons from the reduced NADH plus H and passes those electrons to the oxidized form of the flavin mononucleotide, it would become reduced. And it takes these hydrogens and electrons onto this nitrogen that loses its double bond, and this is where it becomes the reduced form of FMNH2. So the reduced form of FMNH2 will then pass those electrons to the iron-sulfur center. And the iron-sulfur center, basically iron acts as in its oxidized state, it's Fe3+, and it can become reduced as the oxidized form goes to Fe2+. And this is a iron-sulfur center. These are the irons and the sulfurs. And again, it goes from its ferric to its ferrous state as it becomes from oxidized to being reduced. So the electrons are passed from the NADH to FMN to the iron-sulfur center and then to the ubiquinone. Now, as these electrons pass through complex 1, it is said to have undergone a conformational change where it will allow this complex to pick up these protons, these hydrogen ions, and pump them through this protein channel to the intermembrane space. So there, for these two electrons, for one NADH, the two electrons coming from there, complex 1 will promote 4 hydrogen ions to the intermembrane space. Now, this is a picture of ubiquinone. This is a quinone ring and a long isoprenyl tail. And this is one isoprenyl unit, and there are 10 of these on ubiquinone. And this is its oxidized form, the reduced form. The hydrogens, the electrons, and the hydrogens will bond to this double bonded oxygen, thereby reducing it to ubiquinone. It's QH2. Then these two electrons will pass through, and this long tail is hydrophobic and allows it to work through this lipid bilayer of the inner membrane of the mitochondria. So it will transfer, and this QH2 is mobile, whereas each of these complexes, 1, 2, 3, and 4, are stationary. Now, ubiquinone and cytochrome C are mobile. Ubiquinone transports the electrons within this lipid bilayer to cytochrome B. And let's see how it does that. It will transport it to cytochrome B, and this is at cytochrome B. It is a heme molecule, the same as hemoglobin and myoglobin. And if you notice up top, it has double bonded carbons. And so as these electrons will be passed from the initial oxidized form of cytochrome B, then those electrons will reduce that, and then we'll pass those electrons to the oxidized iron-sulfur center from its ferric oxidized form to ferrous state iron, and then it will pass those electrons to cytochrome C1. And this is a picture of cytochrome C1. As you notice, the double bonded carbons now have a sulfur molecule there, and this has more reduction potential than the iron-sulfur center had. So it will gain the electrons to the cytochrome C1, and this is also the same as the cytochrome C. So these electrons will be transported through this complex 3, and as it does this, it changes the conformation of this protein complex, where it will transport 4 hydrogen ions into the membrane space from the matrix. So it will deposit the 2 electrons to the complex 4 as it is passed from cytochrome C1 to cytochrome C, and this is on the outer side of the inner membrane, and it transports those to complex 4. Those 2 electrons will be deposited to a copper molecule in complex 4 from its oxidized form to where it is reduced, and then we'll pass those electrons to the oxidized form of cytochrome A. And this is cytochrome A. It's a little different from the cytochrome B and cytochrome C. This is cytochrome A, where it has this long isoprenyl tail. And the electrons will then reduce this cytochrome A, and then it will pass those electrons to this bimetallic cytochrome A3 to a copper B. They're kind of both joined together. We'll pass those electrons down to here, and then those 2 electrons will bind with one of the oxygen units. This is half of a diatomic oxygen, O2, and so therefore half of this is one oxygen. Those 2 electrons will bind with that one oxygen, and as those electrons passed through complex 4, again, it did a conformational change and allowed 2 hydrogen ions to go into the intermembrane space. And then the other 2 hydrogen ions will come down here, bind with the oxygen, the 2 electrons, with the 2 hydrogens to promote a molecule of water. Now that is from the reduced form of NADH plus H. Now from succinate, an intermediate in the tricarboxylic acid cycle, it would become oxidized to fumarate. This is succinate, and as we lose 2 of these electrons and 2 hydrogens, it will be converted to fumarate. Those 2 electrons will be accepted by the oxidized form of flavin adenine dinucleotide to its reduced form. This is the flavin adenine dinucleotide. Here is the isoalloxazine ring, the 5-carbon sugar, its phosphate, and another nucleotide, the adenine nitrogenous base, its 5-carbon sugar ribose, and another phosphate. So this is the oxidized form, and the succinate will transfer those 2 electrons to this oxidized form where the nitrogens will pick up those hydrogens again, becoming its reduced form of flavin adenine dinucleotide, H2, and those electrons then will be passed to the iron sulfur center. This complex 2 has 4 subunits, succinate dehydrogenase A, B, C, and D. So the electrons will then be passed through here to ubiquinone again. And then from ubiquinone, it will be passed, as it did with the reduced form of nicotinamide adenine dinucleotide, it will be passed from ubiquinone to complex 3 to the cytochrome C to the complex 4, and then to promote another molecule of water. So here we go with the 2 electrons will be transported to the ubiquinone, and then this is the reduced form of the flavin adenine dinucleotide. So let's focus our attention now on the ATP synthase. These hydrogen ions, there are, for every 1 nicotinamide adenine dinucleotide in its reduced form, will promote 10 hydrogen ions into the intermembrane space. And this creates a pH gradient and also an electrochemical gradient, promoting a potential energy. So that potential energy, and this is the, as we transfer these electrons through and we pump these hydrogen ions across, that is the chemiosmotic theory. Now we have an energy potential up here where we can transfer these electrons through the ATP synthase to promote an adenosine triphosphate. Now, ATP synthase, this complex, is the head of it is F0, and this will rotate to whereas when these hydrogen ions come through, it will cause this to rotate, and this is like an electrical generator. It will cause an energy to, as this gamma stalk will help this F1 complex undergo a conformational change where it will accept the adenosine diphosphate and a phosphate on complex B, the subunit of F1, to convert it to an adenosine triphosphate. So for every 3 hydrogen ions that go through, the ATP synthase, it will yield 1 adenosine triphosphate. So as we go through this matrix space, and it creates a conformational change through this F1, thereby allowing the adenosine diphosphate and another phosphate to bind to the beta subunit, creating an adenosine triphosphate. Now this is the oxidation phosphorylation. The oxidation is where each one of these protein complexes become oxidated, thereby creating an adenosine triphosphate, the phosphorylation. Oxidation, phosphorylation. So for every 1 nicotinamide adenine dinucleotide in its reduced form, that will yield 10 hydrogen ions. And for every succinate that is oxidized, that will yield 6 hydrogen ions because it doesn't go through complex 1, it goes to ubiquinone. complex 3, C, complex 4. And so you get 4 hydrogen ions pumped through complex 3 and 2 pumped through complex 4. Now, that is why the NADH plus H will yield 3 ATPs, whereas the flavin adenine dinucleotide will only yield 2 ATPs because it has 4 plus 2 is 6 hydrogen ions. This is energy. Once the triglycerides are broken down, the fatty acids are in the cytosol, but we need to get it into the mitochondrial matrix. In order to do that, we have to activate it. And let's follow this palmitic acid, palmitate, a 16-carbon fatty acid. Let's follow it from the fatty acid to acetyl-CoA. And this is beta oxidation. So in order to get this fatty acid into the matrix, first, like I said, we're going to have to activate it. And so it's going to take a coenzyme A. And the coenzyme A, in order to attach to this fatty acid, is going to take 2 ATPs. Now, once it has been activated, it will go to the mitochondrial membrane, and this coenzyme A will be substituted for carnitine ACL transferase that will transport this fatty acid across the membrane and then into the mitochondria. Once it's in the mitochondria, this carnitine ACL transferase 2 will take off the carnitine off of this and put the coenzyme A back on it. So then, let's follow that as that works. So now we have the coenzyme A attached to this fatty acid, and now it is a 16-carbon fatty acyl-CoA. And let's do some beta oxidation with this. This is the alpha and beta carbon. The reason it's called alpha-beta carbon is because this carbon here is a carbonyl. It is the carbon that's attached, double bonded to oxygen. So that is the carbonyl, and this is the alpha and beta carbon attached to that. It's the first and second carbon. Now what we're going to do eventually, in the long run, what we're going to do, the end result here is we're going to break this bond, this beta oxidation. We're going to oxidize this bond, creating this 2-carbon acetyl-CoA. So let's follow how we do that. First, we're going to use flavin adenine dinucleotide in its oxidized form, and it will be reduced as we take a hydrogen and electrons off of the beta carbon, and then it will be reduced even further as we take another hydrogen off the alpha carbon. Then, by taking those electrons off, we're going to end up with the electrons here forming a double bond. Now what we're going to do is we're going to hydrate that double bond. First we went through oxidation. Now we're going to do hydration. We're going to hydrate that double bond. And so now we're going to end up with a hydroxyl and a hydrogen across that double bond, creating the single bond again. Now we're going to do another oxidation process with nicotinamide adenine dinucleotide in its oxidized form, and it will reduce this fatty acyl-CoA here by taking a hydrogen off of each side of that alpha and beta carbon, thereby reducing the NADH to NAD positive to NADH plus H. Now the electrons will kind of reconfigure themselves because of the cation, and we'll end up with a double bonded oxygen. Now what we're going to do is another coenzyme A is going to and they go through a process of thiolysis where this bond will be broken here and the coenzyme A will attach there. Now what we have done is oxidation, hydration, oxidation, and thiolysis by putting this coenzyme A onto this. Now this is a 14-carbon fatty acyl-CoA as we oxidize 2 carbons off of that 16-carbon fatty acid, fatty acyl-CoA. So this is how we get 1 acetyl-CoA, and this was 16 carbon long, and we're going to do that for every 2 carbons down this fatty acid. And that will increase the number of acetyl-CoAs. And you'll see why we do this later in a couple more slides of why we need this energy, why we do this beta oxidation. And that is just one. So the end result of this 16-carbon fatty acid is there are 8 acetyl-CoAs. Since it's 16, split that in half because we're taking 2 out of it at a time. So that's 8 acetyl-CoAs. We get 12 ATPs for one acetyl-CoA. So 8 times 12 is 96. Now, we only need 7 rounds of oxidation, beta oxidation, because the last 2 carbons are simply not going to, they're not going to have to be oxidized. The CoA, the coenzyme A will just attach there, and we won't need that last oxidation process. So there are 7 rounds of oxidation, and so that will yield, it takes 7 FADH2s, 7 NADHs, it will yield, and for every FADH2, we get 2 ATPs, and for every NADHs, we get 3 ATPs, so that is 1421, which will give us 131 ATPs. And the beginning, we had to activate this, so that was 2 ATPs, and we get a net gain of 129 ATPs for the beta oxidation of a 16 carbon fatty acid palmitate. Now, this is in comparison to a 6 carbon molecule of glucose, which will yield 32 ATPs. So this, you get a much more energy from adenosine triphosphate than you would carbohydrate. But carbohydrates are needed specifically for specific tissues, and I'll show you that coming up. So amino acids as energy. We have many different amino acids coming in the citric acid cycle. Actually, they will come into pyruvate and come into acetyl-CoA, and they will come into the citric acid cycle at different points. What I'm going to point out here is the difference between ketogenic and glucogenic amino acids. Amino acids for energy, they will yield glucose and lipids. So we have the amino acids, asparagine, these in yellow are glucogenic, means that they will make glucose. They can make glucose. They can go around the citric acid cycle and go to the electron transport chain, or they can come back up to oxaloacetate, make PEP, phosphenylpyruvate, and then make glucose through gluconeogenesis. Again, that's not a reverse of glycolysis. So these amino acids, asparagine, aspartate, phenylalanine, tyrosine, all these will come into different intermediates on the citric acid cycle. And then once they come in, these yellows are glucogenic. They will come in here at alpha-ketoglutarate, succinyl-CoA, fumarase, oxaloacetate, and go into phosphenylpyruvate to glucose. Or some of them can come into pyruvate and go into oxaloacetate and participate in the citric acid cycle, the TCA cycle. Or they can come into pyruvate, go to acetyl-CoA, and go to fatty acids. Now, there are some of these amino acids like leucine and lysine that are specifically ketogenic. They will not participate in making glucose. They are strictly, they can come down to leucine and isoleucine and tryptophan will enter into acetyl-CoA, and they can thereby go into fatty acids, 2 fatty acids being built at a time to build a fatty acid for fat storage. Or these amino acids can come into the citric acid cycle, again, and participate in electron carriers and hydrogens going to the electron transport chain. Now, leucine, lysine, phenylalanine, and tryptophan will come into acetoacetyl-CoA, which can go back and forth either to acetyl-CoA, both of these are interchangeable, and they can go into fat storage, or they can come into the citric acid cycle, again, to participate in the NADHs and FADHs to go to the electron transport chain. So basically what we're looking at here is 2 different ways, 2 different types of amino acids, glucogenic and ketogenic, where some of these amino acids will go to acetyl-CoA for fat storage, or the electron, the TCA cycle, and into the electron transport chain. Or the glucogenic amino acids can be made into glucose through gluconeogenesis. So that's how amino acids are used for energy, making glucose or lipids. Now, energy balance. I'm going to briefly go over energy balance here, but in the next presentation, that will be more focused on energy balance and body composition. So here, energy balance, according to the U.S. dietary guidelines, we're going to use 55% carbohydrates, 15% protein, 30% fat. And, you know, if you want to eat more protein and less carbohydrates, you're going to start altering your metabolism, the way your body functions. And then also you can have excesses. You can eat too many of all of these and not eat enough of these. So we're going to look at excesses and deficiencies in the energy balance here. Excuse me. Excessive nutrient intake. When excessive intake of carbohydrates, proteins, and fat exceeds the body needs, the result is weight gain. Carbohydrates, once glucose needs are met, then the glycogen stores are filled and the excess are costly stored. So we'll start storing the carbohydrates as glycogen, and then the fat, since fat costs little energy to store, excesses are quickly stored as triglycerides. It doesn't take that much energy to store fat as fat. In protein, the body cannot store protein as protein. Daily losses are replenished of the protein losses, and then excesses are oxidized to maintain the nitrogen balance, as we learned earlier in the protein lecture. The carbon skeleton of excessive protein intake are then stored as triglycerides in adipose tissue. So if we have too much energy consumption and proteins are also in excess, they will be stored as fat or excreted. The nitrogen will be excreted through urine, and the carbon skeletons will then be stored as fat. As we saw just a minute ago, the amino acids that are ketogenic. So inadequate nutrient intake. In times of inadequate nutrient intake, carbohydrates, protein, and fats will eventually be used for energy, each one of them. Glucose, within the first 12 to 24 hours, the glycogen reserves are used up, causing low blood glucose levels. Low blood glucose levels lead to the breakdown of fats and proteins. And then the protein, the first few days of fast, after glycogen stores have been depleted, the brain red blood cells and the nerve cells use oxidized protein and glycerol to meet their carbohydrate needs. And in a prolonged fast, protein degradation is limited to maintain the functions to live. Now, after a few days, like I said, the proteins will start making glucogenic process to make glucose, because the brain red blood cells and the nerve cells need that glucose. They prefer glucose. And then after a prolonged period, after 12 to 24 hours of fasting, fatty acids are the body's primary fuel source, and proteins are used for cells that primarily use glucose. In a prolonged fast, the brain begins to use ketone bodies for fuel, and other cells use fatty acids with sparing protein degradation. So ketosis, after about 24 hours, ketone bodies are the primary fuel for most of the cells. And what a ketone body is, first, when you use your glycogen reserves as glucose, and then you are in a prolonged fast, now you're going to try to use your glycerol backbones from the fatty acids, those three carbons there, you're going to use those as a carbohydrate fuel source. You can't use fatty acids as a carbohydrate fuel source because they break down to acetyl-CoAs and go into the electron transport chain, the tricarboxylic acid cycle, and then the electron transport chain. So this is acetyl-CoA, and you will bind two of these acetyl-CoAs together to form acetoacetyl-CoA. And then we'll lose the acetyl-CoA off of this, and this will be turned into HMG-CoA, hydroxymethyl-gluteryl-CoA, and then another acetyl-CoA will come off. And that will form a ketone body. So all these acetyl-CoAs that are starting to accumulate, they will start building together and form ketone bodies. And this is acetoacetate. This is a true ketone body. And this is actually acidic. And this is where you start to decrease the blood pH. And this, then again, the reduced form of NADH will be oxidized, reducing this acetoacetate to beta-hydroxybutyrate. And this is not a true ketone, but the brain will accept both of these to, it will cross these ketone bodies are able to cross the blood-brain barrier. and use it as fuel. So this is in a prolonged state of fasting. There becomes an overlap of fuel source, ketone bodies as glucose for the brain and the nervous system, as some areas of the brain and red blood cells use only glucose. As keto acid concentrations in the blood increase, pH decreases, and this is what they call ketosis. Ketosis promotes a loss of appetite to conserve energy output. It does that through hormones, and at this point protein is still lost, decreasing lean tissue and decreasing the energy needs. So as you're going through this long, prolonged fast, you're going to start losing lean tissue because the proteins are breaking down to supply specifically red blood cells that will only operate with the carbohydrate source. And other parts of the brain and the nervous system, nerve cells that need glucose itself. So then fasting leads to weight loss at the expense of lean tissue. You start losing muscle tissue, and in a prolonged state, you'll start to lose tissue from organs and structures. So a low kilocalorie diet and activity leads to weight loss, sparing lean tissues, and this is most favorable, and this is what we'll go over in the next lecture.